Good morning. In this video, we will discuss about HDL metabolism. This HDL is synthesized partly in liver and also partly in intestine. Some HDL molecules are synthesized from liver and some from intestine. So this HDL, its function is exactly opposite to that of LDL molecule. The function of LDL is to transport cholesterol from liver to peripheral tissues, whereas this HDL takes the cholesterol back from the peripheral tissues to the liver for its excretion, which is called as reverse cholesterol transport. Since they have contrasting functions, they also have contrasting apolipoproteins. In LDL, we have only ApoB100, but whereas here we have all the other apoproteins other than ApoB. That means ApoA, ApoC and ApoE. Apart from helping in reverse cholesterol transport, HDL also plays an important role in chylomicron as well as VLDL metabolism. By acting as a storage reserve of ApoC and ApoE, it donates the ApoC and ApoE for the chylomicrons and VLDL and help in their metabolism. This is a brief introduction about HDL. Now we will see in detail how this HDL molecule helps in reverse cholesterol transport. As I already mentioned, HDL is synthesized by the joint collaboration of liver and intestine. The newly synthesized HDL, they will be having phospholipids, apolipoproteins A, C and E and very minimal cholesterol. Since the cholesterol content of the newly synthesized HDL is very less, it will be looking like a deflated balloon, so which we call it as discoidal HDL. Okay, the content, nothing is there, only very little cholesterol is there inside, so it will be like a discoidal shape. Okay, so now this discoidal HDL, which is newly synthesized, once it enters into the circulation, it binds to an enzyme called as LCAT, lecithin cholesterol acyl transferase enzyme. It will bind to the LCAT enzyme. After binding, it reaches to the peripheral tissues. Once it comes in close to the peripheral tissues, cholesterol from the peripheral tissues will be transferred to the HDL through any one of these receptors, transporters. ATP binding cassette protein A1, ATP binding cassette protein G1 or scavenger receptor B1. So through these receptors, the cholesterol molecules will be transferred to the HDL molecule. As the cholesterol enters into the HDL, ApoA1, which is present on the surface of HDL molecule, will activate this LCAT enzyme. Okay, so as the name says, lecithin cholesterol acyl transferase, they will be transferring a PUFA molecule, polyunsaturated fatty acid, from lecithin to cholesterol to form cholesterol esters. So the incoming cholesterol will be converted to cholesterol esters by this LCAT enzyme. Okay, so this will facilitate for accommodation of more and more cholesterol molecules within the HDL molecule. So as more and more cholesterol esters are stored within the HDL, the size of the HDL will slowly grow up and it forms a HDL3 molecule. Now this HDL3 in turn again goes to the peripheral tissues, will take further and further cholesterol esters molecule and it will be converted into fully spherical HDL2 molecule. Now this fully spherical HDL2 molecule cannot take up further cholesterol esters. Why means the cholesterol esters, the already fully loaded cholesterol esters, by feedback inhibition, they will inhibit this LCAT enzyme so that further cholesterol cannot be stored within it. Okay, so in order to relieve this feedback inhibition, HDL2 what they will do means they will donate some of its cholesterol content to the other lipoproteins through an enzyme, through a protein called as cholesterol ester transfer protein. Okay, so this protein actually it helps in exchange of lipids. From HDL2, cholesterol will be given to the other lipoproteins, whereas triglycerides from these lipoproteins will be transferred to HDL2. Okay, so ultimately what is happening means the cholesterol content in the HDL2 will be reduced. So LCAT, the feedback inhibition on LCAT will be removed. So HDL2 can further remove cholesterols from the peripheral tissues. Once it has taken enough amount of cholesterol from the peripheral tissues, the HDL2 will go to the liver. So in the liver, we have a specific receptor for HDL2 called as scavenger receptor B1. So this scavenger receptor B1 identifies the HDL2 molecule with the help of the apoprotein ApoA1. Okay, so the ApoA1 present on HDL molecule has two different functions. One is it activates LCAT and helps in the storage of cholesterol molecule within the HDL. Second, it also acts as a ligand for the hepatic uptake by scavenger receptor B1. Okay, so through this receptor, HDL2 will be bound. Okay, after binding to these receptors, the cholesterol content of the HDL will be sucked up into the liver, whereas the triglyceride contents in the HDL2 will be removed either by hepatic lipase or endothelial lipase. So either by any one of these enzymes, triglycerides will be removed, and through scavenger receptor B1, the HDL, the cholesterol molecule will be removed. Okay, so this delivery of cholesterol from HDL to liver, it can be either partial or it can be complete. Sometimes only some quantity of cholesterol may be delivered, so that what will happen means the HDL2 can be converted again back to HDL3. Now this HDL3 in turn again can go to the peripheral tissues and they can collect further cholesterol molecule. Okay, and they will be forming the HDL2. So this interchange of HDL2 to HDL3 and HDL3 to HDL2 is called as HDL cycle. Okay, so this helps in taking more and more of cholesterol from the peripheral tissues by a single HDL molecule. Alternatively, the HDL2 can donate all its contents into the liver and it can get transformed into pre-beta HDL. That means it will be delivering all the contents and it will become pre-beta HDL, which will be having only the apolipoproteins. So this pre-beta HDL, which is rich in apolipoprotein A1, will then be converted to discoidal HDL, and then the entire process will be continued again. So this is how HDL molecule helps in the transfer of cholesterol from peripheral tissues to the liver. Within the liver, the cholesterol molecule reaching from HDL will be converted to bile acids and bile salts, and they will be excreted through feces. Since HDL helps in decreasing the cholesterol accumulation in the peripheral tissue, since it reduces the body cholesterol level and it favors the cholesterol excretion through the liver, HDL is called as good cholesterol and it reduces the risk of atherosclerosis. That's it about HDL metabolism. Okay, so let me summarize the key points once again. So HDL molecule is initially synthesized with the help of both liver and small intestine. The initially synthesized HDL is called as discoidal HDL. So this discoidal HDL will bind with LCAT and goes to the peripheral tissues. From the peripheral tissues, the cholesterol will be transferred through any one of these transporters, ABCA1, ABCG1 and SRB1 into the discoidal HDL. Okay, so inside the HDL molecule, the cholesterol entering will be converted to cholesterol esters by the enzyme LCAT, which will be activated by ApoA1. Okay, as more and more cholesterol esters are packed into the HDL molecule, the size of the HDL molecule increases to form initially HDL3 and then it forms HDL2. So once it forms HDL2, it will go to the liver and deliver its contents to the liver. Okay, so again this delivery can be either partial or it can be complete. If it is partial, the HDL2 will be converted back to HDL3 and which can further take the further cholesterol molecule from the peripheral tissues. If it is complete, it will be converted to pre-beta HDL, which can again restart the cycle. Okay, so that's it about HDL metabolism. So along with this HDL metabolism, let me discuss about one more minor lipoprotein, which is nothing but lipoprotein A. So this lipoprotein A is made up of apolipoprotein A, which is linked to ApoB100 by a disulfide linkage. So here this ApoA is small a. It is not the Apo capital A which was seen in HDL and chylomicrons, okay, which is used for activating the LCAT enzyme. That is different and this is different. Okay, this is Apo small a. Okay, so this ApoA small a will be bound linked to ApoB100. Along with that, few lipid molecules will also be there. Okay, so this structure of lipoprotein A, it has been found to be structure similar to plasminogen structure. So you all know what is the plasminogen. Plasminogen is the inactive form of plasmin, which when activated by an enzyme will be converted to plasmin, and that plasmin will break the fibrin and leads to clot destruction. This is the normal function of plasminogen. But our lipoprotein A, which is structurally similar to plasminogen, they can go and bind to this activating enzyme, and by competitive inhibition they can prevent this activation process. If this activation is inhibited, plasmin will not be formed, fibrinolysis will not be happening, so it will favor clot formation, which that means it can lead to the risk, it can increase the risk of atherosclerosis. Normally, this lipoprotein A should be less than 30 milligram per deciliter. But if it is more than 30 milligram per deciliter, it can increase the risk of atherosclerosis. Okay, so that's it about lipoprotein A and HDL metabolism. Thank you.